Welcome my friends in another video tutorial from microbiology with Shagnik. So in this video tutorial, as I have promised in our previous video, that we will going to do some different thing. So here today we will going to talk about the pentose phosphate pathway, okay, which is an alternative pathway of glucose oxidation. So without any further delay, let's get started with the video. Okay, so first of all, today I will going to try to upload this video in English language. So if you face any kind of problem in my pronunciation or anything regarding understanding the topic, then you can contact with me via comment box. Okay, so let's get started with the video, friends. First, so pentose phosphate pathway, which is also known as hexose monophosphate shunt or phosphogluconate pathway, is our today's topic of discussion. Okay, so first of all, here oxidation of glucose occur. Not only oxidation of glucose, but here oxidation of glucose 6 phosphate take place. Okay, that is the thing we should remember. Now the enzyme involved in pentose phosphate pathway all are located in either cytosol or in plastids. Okay, so there are two distinct phases that mainly present in pentose phosphate pathway. First phase is our oxidative phase and second phase is non-oxidative phase. So oxidative phase occurs first and non-oxidative phase occurs then. Okay, so in case of oxidative phase, what happens that the glucose 6 phosphate, which is the main substrate for pentose phosphate pathway, will be oxidized. Okay, and there will be generation of ribulose 5 phosphate and 2 moles of NADPH. We will going to see how it occurs. And in case of second phase, which is non-oxidative phase, remember from oxidative phase ribulose 5 phosphate will going to enter in the non-oxidative phase, and from this ribulose 5 phosphate there will be generation of 2 molecule of glucose 6 phosphate and 1 molecule of glyceraldehyde 3 phosphate. Okay, so let's see how the entire thing is going on. So first of all, oxidative phase. So as I have told you that the substrate is glucose 6 phosphate, right, and we will going to form ribulose 5 phosphate. So before going to enter this thing, I want to tell you that this picture I have taken from Pathfinder CSI and Net Life Science Volume 1. So you can also refer this book to understand this. Okay, so what happened first? First of all, there will be oxidation of glucose 6 phosphate. Okay, in presence of NADP plus. Remember that in case of glycolytic cycle, NAD plus was involved, right? And in case of this pentose phosphate pathway, we will going to see the case of NADP plus. Okay, so enzyme involved in this entire thing is glucose 6 phosphate dehydrogenase, which will convert glucose 6 phosphate into 6 phosphogluconolactone. Okay, then there will be a hydrolysis reaction in presence of water and in presence of enzyme gluconolactonase, which will convert 6 phosphogluconolactone into 6 phosphogluconate. Okay, then there will be oxidation step in presence of NADP plus again, and not only this is oxidation, but it is oxidative decarboxylation with removal of CO2. Okay, that will give us from 6 phosphogluconate ribulose 5 phosphate. Okay, in presence of enzyme 6 phosphogluconate dehydrogenase. That is the entire thing of oxidative phase. Now there is couple of things that we should remember from oxidative phase. First of all, there will be generation of 2 moles of NADPH, and there will be also generation of 1 mole of CO2. Okay, and in oxidative phase there is too much thing present that is different from glycolytic cycle. First thing is, as I have already mentioned, that NADP plus is involved in case of oxidative phase of pentose phosphate pathway, but in case of our glycolytic cycle there is NAD plus. Okay, and in case of pentose phosphate pathway there is production of CO2, as you can see, but in case of glycolytic cycle there is no CO2 production, right? And in case of glycolytic cycle, ATP production was a very important thing, right? We should say that in case of glycolytic cycle, ATP production was the main thing, right? But in case of hexose monophosphation, as you can see, there is no direct ATP production. Okay, so that is the idea of our oxidative phase. Now there is a regulation in oxidative phase, that is, in case of oxidative phase, the committed step is this one. Okay, if it happens, then the entire thing will surely be completed, right? That is the concept of committed step. If committed step happens, then the entire step will occur, right? So committed step can be regulated by concentration of NADP plus. Just consider a situation: if cell consumes NADPH, then the concentration of NADP plus will rise. Okay, and this increased concentration of NADP plus will increase the activity of glucose 6 phosphate dehydrogenase, facilitating the conversion of glucose 6 phosphate into 6 phosphogluconolactone. That is the thing. So that is the things we should remember from our oxidative phase of pentose phosphate pathway. Okay, so that was the first video from our pentose phosphate pathway things. So today we have talked about our oxidative phase. In the next video we will going to talk about non-oxidative phase. Then there will another video for functions of pentose phosphate pathway, and at last we will going to talk about some deficiencies. Okay, I have decided to divide the entire pentose phosphate pathway thing into 4 different videos, okay, for a better understanding of the topic. So stay tuned. Thank you.